So my research team focuses on cervical level injury, which, as most of you will know, is where most spinal cord injuries occur in the human population. And those injuries at that level affect a huge number of motor, sensory, autonomic functions below the level of injury, and obviously functions at the level of injury. And at cervical levels, they compromise the phrenic motor system, which is something I'll show you a little bit about. But that phrenic motor system controls the diaphragm, which is the primary muscle of respiration. And so that impairments to respiration or breathing typically necessitate assisted ventilation, lead to subsequent disorders, and as Dr. Snyder said, they are the leading cause of morbidity and mortality in people with cervical level injuries. So to give you an outline of the way my talk's going to be structured, I do want to open up to the people in the room here. If you have questions as we move through, I'll try and find places where I can pause for additional questions if you'd like to ask questions throughout my talk, make it a little more discussive for people that are interested. But otherwise, this is the general flow of my presentation. I'm going to introduce the models we use to look at cervical level preclinical spinal cord injury and how those models affect respiration. I'm going to briefly describe the types of plasticity that we see after injury with a special focus on spinal interneurons. That's something that you'll hear me keep coming back to throughout my talk, because this is one of the populations we think is not only crucial to better understanding after injury, but we think they represent an ideal population for transplantation as well. And then I'll go into some of the transplant studies we're doing and how those can be used to repair that damaged spinal substrate. So these two schematics here give you an example of, on the left, human cervical spinal cord injury, and on the right, a preclinical animal model. So we use small rodents for our preclinical studies, mice or rats. And it shows you what the cervical spinal cord looks like. These schematics I actually created from fixed tissue specimens from both human and from small animal models. So with human spinal cord injury, it's usually consists of a number of different types of injuries, where you can have initial contusion injury, there can be subsequent additional compression injury to the tissue, with occasionally different types of laceration, which can be due to penetration of bone or other foreign objects. So it's really a complex kind of injury in the human spinal cord injury, which is usually quite heterogeneous between each individual as well. Preclinically, we model aspects of that. So we typically model a partial laceration, like with the hemisection as shown here at the top, or we have types of contusive or compressive type injuries. These examples I'm showing you of the lateral hemisection at a high cervical level or mid-cervical contusion injuries are the types of models that my research team uses and many others do to study how these injuries affect respiration, these respiratory networks. And with that, I'm going to introduce to you what the phrenic motor network actually looks like. So you're looking at a bird's eye view of the spinal cord in the middle with a partial cross-section just to the right of that. And we can break down the different substrates within the phrenic network and how we can identify them. So the first is a population of the phrenic motor neurons, which reside from C3 to C6 in the spinal cord. These motor neurons extend their axons out to innervate the diaphragm on each side of the spinal cord. And these can obviously back labeled and traced with a number of different traces, so we can map with the distribution of these cells. As you can see here, we have a longitudinal section of the spinal cord that's demonstrating the distribution of those motor neurons. We can also look at the descending drive onto this phrenic substrate, which comes from the medulla. The ventral respiratory column sends its axons down directly onto these motor neurons to control their function. In addition, we can use these traces or combine these traces. So you have anterograde tracing from the VRC to see how these descending axons innervate those motor neurons, and we can see how that might change after a spinal cord injury. By using transsynaptic tracing or viral tracing, as my research team uses, we can also map the distribution of spinal interneurons. So as I said, we think these are really important for better understanding how they're connected to the network normally. We know these neurons now exist within the phrenic motor system, regulate phrenic motor function, and these contribute to plasticity after a spinal cord injury. Again, we use a transneuronal viral tracer to back label the distribution of these spinal interneurons. We can also look at primary afferent innervation of this spinal phrenic motor network, using transganglionic tracers that can be applied in the periphery to look at the distribution of these primary afferents and how they innervate these motor neurons and interneurons. And we can look at using immunohistochemistry to look at other populations of descending axons. So really we have a battery now of different anatomical markers which we can very comprehensively assess what this phrenic motor network in the cervical spinal cord looks like in a naive animal, how it's affected by a spinal cord injury, and how it might change with subsequent treatments. And this is just showing you the innovation of the serotonergic axons into the cervical spinal cord normally and how they innovate that ventral horn and motor neuron pool. So now we can also then piggyback off this well-identified anatomy to start looking at functional changes within this respiratory system. And the first way to start assessing respiration is by using whole-body plethysmography, which is something we do clinically, or we can use spirometry to essentially measure different aspects of ventilation. Now, ventilation isn't just phrenic function, it's a function of all respiratory systems, so the phrenic network, the intercostal networks, and even the abdominal networks, and how they all contribute to each breath. So the breathing frequency, the tidal volume, and therefore the minute ventilation. So we can affect these ventilatory parameters and see how they change after injury. But to look at how the phrenic motor system itself functions, we use electrophysiology. And we can record directly from the phrenic motor output to look at the phrenic nerve recordings, or we can place electrodes in the muscle, the diaphragm itself, to see how diaphragm activity changes after injury. And by combining these functional outcome measures, we can see how diaphragm activity contributes to each one of these ventilatory parameters and how that changes with injury, disease, or with subsequent treatments. So we know that by studying these different aspects of the phrenic motor system, we can very effectively identify where the phrenic network is, the spinal phrenic network, how it receives input from the brainstem, and how both are compromised by these different types of injuries. Now here I'm showing you a contusive injury which directly affects this population of motor and interneurons and the descending input onto that phrenic substrate. Looking at the function after one of these injuries, we can see that on the side of the injury, the degree of diaphragm activity has decreased. Soon after injury, it's completely paralyzed, but there is a progressive recovery over weeks in nearly every animal model that's been studied. We see this progressive amount of recovery within the muscle, which is attributed to this ongoing spontaneous plasticity. And this phenomenon is also now being detected in the human population, where we've been able to track muscle function over time. You see on the contralateral side that the diaphragm works relatively normally, responds to a respiratory challenge. And here these challenges are elicited by either lowering oxygen or increasing the amount of carbon dioxide in inhaled air, making the respiratory system work harder. And we can see how these networks respond to that increased challenge. But on the side of injury, there's a very limited response to this respiratory challenge. Clinically, that's known as respiratory insufficiency, and it's something we can model quite effectively in our preclinical animal studies. But the focus, once again, is on this population of spinal interneurons. We know they exist and they're connected normally, and we're seeing how they change after injury. And the field over probably the last 20 years in spinal cord research has become really interested in these populations of spinal interneurons and how they can contribute to plasticity. And we know from studies after cervical or even thoracic injury, these interneuronal networks reform new connections to create novel pathways that can facilitate either spontaneous functional improvement to some extent, but that can also be therapeutically driven by activity-based therapies, rehabilitation, and more recently there's extensive evidence to show that implantation of devices and neural stimulation also elicits recovery, attributed largely to these reorganization of these spinal interneuronal networks. So this is a cross-section of the cervical spinal cord, in this case of an adult rat, but it's showing you the complexity of the anatomy within this cervical spinal cord, even in just one thin section of the spinal cord. So what we're actually looking at here in red is a population of spinal neurons in general, all the neurons which are in the middle. In blue, we can start looking at subsets of neurons. So for the most part, we've labeled populations of motor neurons. But you can also start to see subsets of spinal interneurons throughout, especially this part of the spinal cord, which you'll see again and again throughout my talk, where we have populations of spinal interneurons that are again involved with this spinal plasticity after injury that can form new connections. So we and many others believe that spinal interneurons are key to this plasticity after traumatic injury. The question, of course, then, given that there's hundreds, if not thousands, of different types of these spinal interneurons, which interneurons are of interest for recovery and plasticity, and which might represent therapeutic targets. To better understand that, we go back to the developing spinal cord. And what we're learning from developmental neurobiology is that we can track the expression of different transcription factors to start identifying subsets of these spinal interneurons. And shown here in red is a population known as the CHUX10 expressing or VSX2 expressing spinal interneurons, which are considered the V2A population, which is a population of excitatory cells we'll learn more about throughout my talk. But this is an example of the different types of populations of interneuronal progenitors that are distributed throughout the developing embryonic spinal neural tube. Very loosely considered to be, for the most part, in this dorsal area sensory, and this ventral part mostly motor. That's not clear cut. It's not. There's obviously a lot of overlap that we've learnt. But these cardinal classes of interneurons can be used to start categorizing some of these types of interneurons. As we learn more about ways to categorize cells through electrophysiology or single cell sequencing, limiting ourselves to just these cardinal classes might be too limiting. There might be other ways to start classifying these cells, but at least this provides a good template for understanding what types of cells we have and how they might contribute to plasticity. We can even track the distribution of these cells over time to see with maturity through the spinal cord where these cells are and how they connect to different motor, sensory, and autonomic networks, and start to track these cells over time. So a couple of subsets of cells that I'm going to talk about in more detail are the V2A interneurons, the V0 cells, and also the V1. So all these are ventrally isolated and are known to be premotor populations of spinal interneurons. So the V2A spinal interneurons are a population of excitatory cells within the cervical and lumbar networks known to contribute to a wide range of functions. So using electrophysiology and tracing techniques, we know they innervate the locomotor networks, the upper extremity motor networks, and also the respiratory networks. A study we did a few years ago showed that these cells are not only connected to the intact phrenic motor network, as shown here, but they also increase their connectivity spontaneously after a high-level spinal cord injury. As you can see, the number of these cells in these red bars that become spontaneously connected after a spinal cord injury does vary a little bit, but interestingly, the number of cells that connect to the phrenic network after injury directly correlates to the degree of recovery that each one of these animals sees. So there is a correlation between these V2A interneurons and functional recovery. We can also start to look at the cholinergic population of cells, and within the spinal cord there are a few different populations of cholinergic interneurons that are of interest to us. One of these premotor populations around that central area of the spinal cord are the PTX2, is a transcription factor that's been known to identify them, or also known as the V0C population, cholinergic spinal interneurons. We've shown that they are also connected to the phrenic motor network normally, and once again, they increase their connectivity spontaneously after injury, although fairly modestly. This is a significant increase in this connectivity after traumatic injury. So we've become interested in exploring these V0C's interneurons in more detail, much like we have the V2A. And in collaboration with a group in Scotland, Gareth Miles's team, people from both his research team and mine have been pairing up to start looking at how these cholinergic interneurons contribute to plasticity after traumatic injury. We're also working with the Gladstone Institute to use tissue clearing approaches to start mapping the distribution of these interneurons, how they might change after injury, and how these neurons are contributing to functional improvement. The third population of cells I'm going to talk about are the V1 inhibitory spinal interneurons. So again, they're premotor, but they have an inhibitory function. And so in collaboration with Jay Bickoff at St. Jude's, we're using another population of transgenic mice to see how these V1 engrailed 1 expressing interneurons are connected to the phrenic network normally. There are many, as you can see in this cleared tissue here, populations of these V1 interneurons normally. This is a study that's being done by one of my graduate students to map the distribution of these V1 interneurons and how they're connected to the phrenic network. And surprisingly, even though this population of V1 inhibitory cells is the probably the largest number of inhibitory premotor cells in the spinal cord, there aren't a lot of connections to the phrenic network normally, and it doesn't seem to change much after injury, although this is an ongoing study. So with the focus of these V0C cholinergic cells and the V2A interneurons, we know they exist within the uninjured phrenic motor network. We know that the V2A interneurons spontaneously connect and contribute to anatomical and functional plasticity after injury. We know more recently that cholinergic spinal interneurons also contribute to changes in connectivity normally, and that can be even enhanced with interventions like the activity-based therapies. So the question is, how can we go about enhancing these interneurons, targeting them therapeutically? And the way my research team has tackled this is twofold. The first is to be looking at activity-based therapies, like I said, rehabilitation, and the second is using our cell transplant strategies to harness the capacity of these interneurons for repair. I'm just briefly going to touch on some of the rehab strategies or activity-based therapies we can use as an intervention to target these spinal interneurons, because this will come back towards the end as something we want to start combining with cell therapy as well. So there are many ways you can drive neural activity within the spinal cord. As many of you will know, this schematic gives you an idea of some of the ways we can approach that. We typically have broken it up into neural interfacing and mechanisms for providing a device that can facilitate activation of these neural networks, or we can use activity-based therapies. In general circumstances, that can often be considered like locomotor therapy, treadmill training. Within the respiratory system, we use changes in the gas that are inhaled, like I was describing before, to change the way we breathe. So we can either decrease oxygen, increase CO2, and either one of these strategies essentially can form as a way of rehabilitating or driving activity within these respiratory networks. And that's what I'm going to focus on very briefly to talk about how we harness these to tap into our spinal interneuronal networks. We know that these strategies can enhance supraspinal plasticity and drive the spared pathways and how they might contribute to plasticity after injury, but also activating these spinal interneuronal targets, these spinal networks, can drive plasticity. And that's been shown very comprehensively for many people throughout the field of SCI research. So looking at using these respiratory training strategies, this will very briefly give you an idea of how we can use these in preclinical models. So the animals are usually within sealed gas chambers where they have very controlled inhaled gas throughout essentially just resting. So they just sit in the chamber and their exercise simply is breathing the gas we provide them, which is my ideal way of exercising, just sitting there and doing nothing and breathing. So we expose them to either just normal compressed air normally, or eupnic breathing, which is just regular non-trained animals, or we can change the gases they breathe, lowering the oxygen or increasing CO2 to make those respiratory networks work much harder. And we do this in an intermittent way. So we have 5 minutes of training, 5 minutes of rest, and repeatedly, it's almost like doing sets at the gym. These respiratory muscles are working, then resting, and vice versa. And by doing this for a daily basis over several weeks, we train these respiratory muscles and the underlying networks innervating those muscles. And this has been shown by many people. This is work pioneered really by Gordon Mitchell and many others throughout the field of respiratory neurobiology. These approaches have been very effectively used to drive neuroplasticity within these respiratory networks. So we're looking at how these same approaches can be used to tap into these spinal interneurons and how they can contribute to functional improvement. And we've shown that by training, using either intermittent hypoxia here in green or intermittent hypercapnia shown here in orange, we increase the number of these phrenic interneurons that are integrated with our phrenic network. And more specifically, we increase the number here of the number of cholinergic interneurons, so these V0C interneurons and how they're connected to this phrenic motor system post-injury. And these respiratory training approaches increase functional recovery. So that we're seeing in orange here with intermittent hypercapnia, you get a significant improvement in muscle activity in the trained animals. So in summary for now, we know that there is spontaneous plasticity within the injured spinal cord. We know that these changes in plasticity contribute to functional recovery after injury. We know that this can be enhanced with activity-based therapies, and others have shown with neural stimulation as well. And we can essentially optimize these training techniques now to see how different either activity-based therapies or stimulation approaches can target different subpopulations of these interneurons to amplify phrenic motor plasticity after cervical spinal cord injury. So the second aspect of my talk, the rest of my talk today, is going to talk about how we can use strategies for repair, so focusing on cell transplantation. I'm going to pause very briefly in case there's any questions, any burning questions before I move through to cell transplantation.
So this is what you've built is a great tool chest with all the specificities of all the different neurons, and the neurons do the work, the neurons make the connections. But thinking of this as a drug discovery scientist, trying to target each one of those types of neurons is going to be challenging. What do you think about looking at things such as microglia and astrocytes as a target population to help with the neuronal health? And have you gone back in and stained for changes in microglia and astrocytes as these changes to the neurons happen?
That's a great question. So we have not selectively looked at changes in any of the glial populations after these existing interventions or how they might change connectivity. I would guess that these glial changes do facilitate some of the synaptic changes that we're seeing, though I don't know how they would respond to the interventions that I've described. One of the reasons we haven't focused on that is because we've taken a different approach, and that's, as you will see, as we move through these strategies for repair, focusing on these different populations of neurons that we're transplanting, we're seeing how the different glial populations might also be necessary in transplantation studies to facilitate some of these ongoing repair strategies. How they contribute to plasticity, though, isn't something that we've had the capacity to look at. Great question. Yeah, just from the stuff we've talked about already, I think there's a number of questions that I simply don't have the capacity to begin addressing, but there's many things. If you're interested in collaborations, feel free to reach out to me. So for looking at using cell strategies to enhance spinal cord repair, for many, many decades now, the last 30, 40 years, people have been looking at a range of different cell types that can be used for neural repair, whether that's to promote repair above the level of spinal cord injury, at the level of injury itself, or even below these denervated networks in the spinal cord, below a spinal cord injury. And the cells that are used have different aspects for repair, whether it's to promote remyelination, change the gliosis that occurs at the level of the injury and see if that can facilitate a degree of repair, or even reorganize the cytoarchitecture by providing new cells that can contribute to new glial populations, new neuronal populations. And for the most part, my work has focused on the transplantation of neuronal or neural progenitor populations and how they can facilitate different aspects of these types of repair. So when we look at the spinal cord, we try and think of basically what are the components that are there that we need to recapitulate this damaged spinal cord and restore some of these elements that are otherwise lost that might be pro-reparative. And so this section of a spinal cord has been stained for neuron in red, for astrocytes in blue, and for endothelial cells in white. And that's because these are just three of the elements we think are kind of important for promoting neural tissue repair. As you can see here, these three elements have become a focus for the types of cells that we've begun transplanting. So transplanting neuronal progenitors with a key focus on what these interneurons are, transplanting pro-reparative glial precursor cells, and transplanting endothelial progenitor cells. The focus of most of what I'm going to be talking about just briefly now is going to be on the first population, these neuronal progenitors, where we get them from and what they might become after we transplant them. So this work really builds on, as I said, decades worth of work looking at transplanting neural progenitor populations. So neural meaning neuronal and glial, and that really comes from embryonic tissue transplantation studies, where we have the developing spinal cord. And again, here we're looking at the cardinal classes of interneurons, but there's also these populations, these tissues contain glial elements, endothelial elements, extracellular matrix, all the things you would need for transplantation. So early on, people were just taking these tissues and transplanting developing embryonic tissues. More recently, people have begun to transplant not just tissues itself, but dissociated, mechanically or chemically dissociated embryonic tissues for transplantation, or piggybacking off what we have learnt using stem cells, either embryonic stem cells or iPS cells, to start creating these populations of neurons and transplanting them after injury. The populations of neurons that we've become interested in, once again, are these excitatory V2A neurons, the V0C neurons, and also the V1 inhibitory neurons, and how they might exist within these transplanted tissues. So this schematic, very crudely, is showing you what happens when you have a disrupted spinal cord, and when we transplant these developing neural tissues into the injury site. We essentially want to use this transplant to create new connections and promote repair. These cells can be transplanted with fluorescent tags so we can see where they are and how they integrate with the surrounding tissue. This is from one of my studies where we did transplant embryonic tissues into the contusion site of a cervical injured spinal cord. And you can see these tissues fill the lesion cavity and give us populations of neurons in red and astrocytes in white. We can also start to see the extension of neurites from these neurons, and this is something that many people have shown, not just my research team, where they extend neurites both rostrally and caudally throughout the injured spinal cord. We've shown with transsynaptic tracing that these cells also innervate damaged networks, and in our case, the phrenic motor network receives input. As you can see here, phrenic motor neurons in red receiving green neurites from our donor cells, and spinal interneurons as well in red, also receiving axons from these donor cells. We can look within the transplant itself and see that in red with our transneuronal tracer, some of these donor neurons have been labeled with our virus, which means they are synaptically integrated with our host network that we've traced, and subpopulations of these donor cells are even cholinergic and could represent this V0C population of interest. We can look at our interrograde tracing to see how these transplants receive input from the brain and brainstem, and these networks that can essentially regulate the way that these cells will work after an injury. Likewise, we can look at primary afferent innervation of these donor cells. And as I said, many people have now shown over several decades, from Paul Ryer's work, John Halley, Mark Dzinski, Itzhak Fisher, they've all shown that these donor cells survive and integrate with the injured spinal cord networks, and they can improve functional outcome. But the degree of recovery is usually incomplete, and from a lot of the studies that have been done over time across different research teams, the degree of recovery in different systems can also be relatively inconsistent. We believe that that is partially because we need to better understand the phenotypes of the cells that we're putting in, and how that those different phenotypes may vary with different populations of cells that we've used to get these neuroprogenitor cells, and what they do once they're transplanted within the injured cervical or thoracic networks. So with a focus once again on spinal interneurons, we know they're key components of plasticity. They can therapeutically be harnessed. We believe that they also represent an ideal cell type for transplantation. So again, we've become interested in which types of these interneurons can be used, and how they might exist within the tissues that have been used in the past, and how we can build upon those early studies. So the two key strategies that my research team has been using for cell transplantation is better identifying the donor cell types we're putting in. And to do that, we looked back at essentially what exists normally within this developing spinal cord, and how that changes when we isolate those tissues and prepare them for transplantation. So we remove the developing spinal cord, we can dissociate them, we can culture them, and each one of these different things that we do to these cells to prepare them for transplantation can affect their phenotype. So here, just looking at the expression of a few different transcription factors which focus on these populations of cells of interest to us, the V0, V1, and V2A interneurons. What we've shown is that when you isolate these cells for transplantation, there's a significant decrease in expression of transcription factors associated with the V0 and the V2A population of cells, which are a key interest to us because they contribute to plasticity within the phrenic motor system. So we're potentially losing the cells of interest when we transplant our developing NPCs. So the next question we had was, how do we go about restoring that? So if we're losing the cells of interest in our NPCs obtained from tissues, can we use stem cells to now engineer new populations of V0 cells and new populations of V2A neurons and transplant those alongside our NPCs? So we're re-enriching the tissue that might be losing V2As, and we can put more V2A cells into our transplants. And that's exactly the results you're looking at here. I'm just showing the diaphragm recovery from these animals. And what we can see is that in vehicle-treated animals, there's a little bit of plasticity, normally after these cervical level injuries, contusion injuries. If we transplant NPCs, so mechanically dissociated embryonic tissues that are transplanted into the lesion cavity, we do get a degree of phrenic motor recovery and diaphragm activity. But if we add V2A neurons to those NPCs, enriched with V2A interneurons, we get even greater recovery still, although with some variability. But there is a significant improvement when we enrich those cells with our V2A neurons. The contribution of these V2A neurons has been further confirmed by additional engineering, where we added an inhibitory dread receptor, the HM40I receptor, to these V2As, and using clozapine, we can deactivate the cells months after transplantation and see the contribution of these V2A neurons to this diaphragm activity. So when we add clozapine to animals that have these V2A transplants, we decrease the degree of recovery, showing that the V2A neurons have been contributing to the extent of recovery in these animals. So as I showed you, we have become very focused on the types of cells we can start be engineering for these cell transplants, and they do contribute to repair. But even with that V2A enriched NPC transplant, there was a degree of variability, right? So if that represents a degree of inconsistency, that's another target for us. We want to get the best recovery every time achievable. How do we go about doing that? And how do we ensure that the right types of cells are connecting to the network of interest? That's where we've begun looking at different strategies for now improving and preserving connectivity of our donor neurons after injury. And so to do that, we've piggybacked off our earlier studies using activity-based therapies to enhance connectivity. We know it enhances plasticity and connectivity within the injured networks normally. Can we use these same strategies to enhance connectivity of our new networks that we've created with our cell transplants? This is work that's been done by Adam Hall, a graduate student on my team, in collaboration with Gladstone. And that's looking at the transplantation of NPCs in combination now with activity-based therapies. So we can map the number of cells that are connected with our donor transplants and how they change with training. These have actually been, what you're looking at here, is the number of PRV interneurons within the transplant. And we can see that just by giving intermittent hypoxia training to these animals for 4 weeks, we significantly enhance the number of these donor cells that become connected to these cells. And we think this has been attributed to change in growth factor expression that also follows on from these activity-based therapy types of interventions. This is just one way we can go about enhancing connectivity, and we're exploring many others. So in summary, we know that spinal interneurons mediate plasticity after injury. That can be enhanced with activity-based therapies. Transplanting spinal interneurons can also facilitate improvements in phrenic motor recovery and diaphragm activity after injury. And by pairing both transplantation and activity-based therapies together, we're increasing the connectivity of these donor cells with our injured phrenic networks. So as we move forward, and we've found new ways to start creating populations of spinal interneurons, we've moved into human stem cell populations to either from embryonic stem cells or for the most part, iPS, human iPS cells, in collaboration once again with Gladstone, to look at creating human V2A interneurons, transplanting those into the injured spinal cord, and seeing if just V2A alone result in any changes in the phrenic motor network. And what we're seeing here is some results from an ongoing collaboration with Gladstone, where we've transplanted a human V2A neurons along with lanusolideva into the injured cervical spinal cord. And we can see how many of these cells become connected over time. We can also engineer these human iPS cells to contain a channelrhodopsin 2, so we can use optogenetics to drive the activity within these donor populations of cells after transplantation. Using pseudorabies virus, we can look to show that some of these cells are synaptically integrated. So these human V2A neurons do become synaptically integrated with our injured phrenic motor network. And by shining blue light onto these transplants, we can also enhance the activity at the diaphragm. So even just by activating the V2A neurons that are transplanted with our injured cavity, which we know is synaptically integrated with the phrenic network, we see significant enhancement of phrenic motor output. I apologize, that color is projecting atrociously. Hopefully those people on Zoom can see that a little more clearly. But what we're seeing here is that a significant increase in that diaphragm activity with optogenetic stimulation of the transplanted cells. So in an ongoing collaboration with Shelley Sakayama-Elberts at University of Washington and Gladstone, we're now looking at how different populations of stem cells compare to what has been done in the past by isolating developing fetal tissue, how all of these populations of cells can be used to create neural progenitor cells, and how we can see changes in the expression of different genes within these populations before or after transplantation. And what that essentially gives us is a better read on how these different cell populations will give us the most consistent types of cells that we can use for transplantation and repair. So building on these studies, which are really focused on just the neuronal populations of cells, we also wanted to explore what other non-neuronal populations of cells how they can be used for repair of the injured spinal cord. And the first thing we wanted to look at was transplantation of the glial restricted progenitors. So looking at these early glial populations that are known to exist within the developing spinal cord, and work done by several people, including Itzhak Fisher, has shown the transplantation of glial restricted progenitors alone can serve as a substrate for repair of the damaged spinal cord. They essentially can change what's happening in terms of gliofibrosis at the level of the injury. They can also provide a substrate that can facilitate degrees of repair across that damaged spinal cord. And this is a study that's been led by a postdoc in my lab and a ongoing collaboration with Shane Lidlow, who's a past colleague of mine from Australia. So by isolating these glial-restricted progenitors, we can firstly look at how they develop within carefully controlled culture conditions, what types of glial progenitors we have. We've used genetic techniques to start screening these cells to see what they are. I was expecting these cells to be all pro-reparative because they certainly have pro-reparative properties, but they are not. They're a mix of reparative and non-reparative types of glial cells, which I guess should not be too surprising. We've also used these cells to engineer a subset of glial cells that we can manipulate later on after transplantation. So what we've done is we've engineered these glial-restricted progenitors with a lentivirus to express either ASCL1 or MIRS, microRNAs and NeuroD1 using TET-on systems. So we can use doxycycline administration into these animals well after transplantation of glial restricted progenitors into the spinal cord to promote the expression of these factors, which will reprogram the cells directly into becoming neurons. So we can transplant glia and at a later time point turn those glia into neurons. The cool thing about this approach is we know that glial cells are reparative when we transplant them after spinal cord injury. They can provide a degree of repair by just altering the gliotic scar. They may also provide a substrate for repair across an injury site, but then months later, we're left with this large area of just glial cells. So what if we could use these reprogramming factors to convert some of them into new populations of neurons? And that's exactly the approach we're using with this strategy. So we transplant the glial cells into the injured cavity, and at a later time point then deliver doxycycline to convert subpopulations of these donor cells into neurons. I'm just showing you here some of the early studies with transplantation of GRPs alone, where the GRPs survive to varying degrees. So we're doing dose-dependent studies to see what populations of donor cells survive an injury. But it does show you, at least for those of you online, you see the spinal cord here with the transplantation of these surviving glial cells within the cavity. And that number of surviving cells certainly increases with dose. And the degree of repair that we're achieving with transplantation of the glial cells also increases as we increase the dose of glial cells. So as I said, these are ongoing studies, and hopefully I'll be able to show you soon after we reprogram these what that does in terms of these new populations of neurons and how they integrate with this damaged phrenic network. The third population of cells we've been working with are endothelial cells, or more specifically endothelial progenitor cells, which is part of an ongoing collaboration with Endocrine Biosciences, where they've been looking at the use of these endothelial progenitor cells, which have been created from populations of human stem cells. And we can transplant these again into the injured cavity. We chose not to do this alone, but rather pair this with our populations of neuronal and glial precursor cells obtained from the developing spinal cord tissue. And a reason we did this was because we found that if you isolate these cells and culture them, we have very clean populations of neuronal and glial progenitors, but we've lost the extracellular matrix, we lose endothelial cells and other populations of cells which could be important for repair. And that's why we wanted to add back in some of these endothelial cells to make sure that after transplantation, we're getting the best degree of vascularity across that injury site, which can promote restoration of neurovascular unit, because certainly surviving cells in the transplant will need vascular supply. With these studies, we've been able to show that we do certainly form new vascular cytoarchitecture across the injury site, comprising some of these human donor ECs, and that our MPCs that we transplant with them do contribute to this neurovascular unit, in this case showing you glial populations wrapping around this blood vessel here. Interestingly, we see massive populations of macrophages, where we actually stained these macrophages, in this case here in blue, that exist within the lesion cavity but do not contribute to a loss of these cells. They actually seem to be happily residing around these newly formed neurovascular units. And when we stain for axons, there are even axons traversing along this neurovascular bridge across the injury site. So in considerations for ongoing work as we move forward, we're interested in what types of other interneurons we can engineer from stem cells to provide new populations of cells for repair, how we can combine neuronal and non-neuronal elements to repair this damaged spinal cord, how we can combine these cell transplant strategies to promote targeted and preserved connectivity, which facilitates functional recovery. And that might be combining either activity-based therapies, or we're also looking at different mechanisms for neural interfacing and neural stimulation. And moving forward, we want to certainly start treating more chronically injured spinal cord as well. And so that's where we're heading in future. And with that, I want to thank some of the people that have contributed to this work, some research members on my own team, graduate students and postdocs, some collaborators that I've had in the Marion Murray Spinal Cord Research Center, and collaborators also around the US, Shelley Sakayama-Elbert, Lana Joladeva and her team at the Gladstone Institutes, Shane Little at NYU, Jay Bickoff at St. Jude's, and also Gareth Miles at St. Andrews University. And with that, I'll take any further questions. Thank you.
Beautiful talk, Michael. A couple of questions about the timing of the transplantation of the 3 different cell populations. Are they done immediately after injury? Do you wait some time before transplanting them? And also related to that, do you know the mechanism through which you're seeing some of this recovery for the GRPs and the V2A populations? Because for the endothelial cells, you showed the increased vasculature, but what about the other 2?
Right, so for the most part, early studies, people used to transplant these cells immediately after injury, where after a contusion injury, we wait for the formation of a cavity. So that cystic cavity that forms 1 to 2 weeks after injury. We've transplanted usually at a week for the majority of studies that I've shown you here. More recently, we've transplanted at 2 weeks, 1 month, and we've looked at transplanting at 3 months later. Some of these more chronic studies are a little harder to do in these small animals where the survival of the animals is not that long either. So we want to look at long-term survival. We can't also look at long-term injury. So it's a bit of a catch-22 in that regard. But we're looking at trying to transplant as late as we can. In terms of the mechanisms for repair, the glia-restricted progenitor cells, we know that they certainly form a substrate along which we get axons growing. We know that these developing glia are definitely pro-reparative. Even studies way back in the 80s by Jerry Silver and George Smith showed that these developing glial cells have a remarkable capacity for being pro-reparative, reorganizing the glial scar, even at an injury site, and facilitating growth of many different types of axons across an injury site. But even that alone is not sufficient for extensive repair, which is why I think we need to look at these other combinations or even conversion of some of these cells to provide new populations of neurons. In terms of the donor neurons, we think it's connectivity. So we see that these donor neurons form new connections with the network we're studying, the phrenic motor system, and that the number of these connections seems to affect the degree of recovery of muscle activity within that diaphragm. So we think we're forming new relays, and certainly the anatomy and functional recordings, electrophysiological recordings, confirm that anatomical bridge across the injury site.
Yeah, so it was nice that you had something for a hematologist and not a neuroscience person. So with the endothelial cells so you showed in some of your models that hypoxia, is sort of one of your stimulants or issues. So in the damaged tissue, is there localized hypoxia, and can you show that the endothelial cells are improving that? Is that thought to be part of this, or is it, as you said, trafficking or repair of other cells as well, what they're doing?
We are specifically looking at the trafficking of other cells and providing a substrate for neural repair. We haven't looked at changes in tissue oxygen saturation. People have historically. There's some work done by Brad Stokes and Paul Ryer, I think it was in the late 80s, that looked at changes in the oxygenation of tissues after a spinal cord injury, and certainly there's a decrease. The oxygen saturation drops significantly, and that probably varies with the type of injury. I think compression injuries where you get greater ischemic damage will give you even greater detriment in terms of oxygen saturation. Restoration, that vascular architecture has been looked at by a few other investigators like Teo Hag in Louisville, Kentucky, showed that restoration of that vascular network after injury is important for restoring oxygen saturation to damaged tissues. Where you have a lesion cavity, there's really no capacity for restoring to such a massive area of tissue loss unless you provide new substrates and new cells in which you can develop a new neurovascular unit, if you like. And I think cell therapy is really probably the only way to go about providing that substrate. Does that answer your question?
Yeah, I mean, but it doesn't sound like you've actually measured it locally.
No.
You could also hypothesize you could just give VEGF or something like that
Right.
could stimulate some endogenous
absolutely.
vasculature, right? If that was sort of a mechanism.
So
here.
that certainly will increase vasculature in the surviving tissue, not within that area of cavitation. So even the combinations of growth factors that people have used or even using scaffolds, doesn't provide really significant enhancement of that vascular site architecture compared with the use of these neural progenitor cells or neural tissues. Seems to get us much better vascularization of that tissue. Measuring the oxygen saturation was really only done by the early studies with Brad Stokes, and I don't recall what effect that had, but I have to look back.
Yeah, Michael, actually I have 3 questions. So the first one's a very simple one. Do you see any connectivity between V3 neurons and the phrenic motor neurons?
We've not looked. I would love to look. So I think the V3 population is a population that hasn't been studied extensively by many people, especially in the spinal cord injury field. And I think that could be a key population, being that they are also excitatory.
Second thing is, so this loss of, the V2A neurons after transplantation, do you have any sense of why they're being lost as opposed to the V1 neurons mechanistically?
None whatsoever. I think it's a really fascinating phenomenon. I think people historically suggested that there seems to be a loss or a selection, if you like, for inhibitory neurons rather than excitatory cells. We don't know why that is. Which one of the methods we are using is resulting in that shift in the population of cells we're transplanting. But these have been very long used, characterized techniques for creating neuroprogenitor cells. So we assume that many people are probably seeing similar patterns of selection for these different neurons, and therefore we need to start considering engineering the cells that are being lost.
And the third one was actually, it's kind of intriguing, using this TET system to basically induce neurons. And so you're driving them with basically with ASCL1 or with NeuroD. So those not only just convert progenitors to neurons, but sometimes they do play a role in converting them to specific types of neurons. So have you looked to see if there's any specificity within those populations?
We are looking. And so we hope that some of them will become V2A neurons. We're looking at trying to find ways to drive them towards becoming V2A neurons even. But we came into a very significant hurdle. That's something we have a paper that's on the bioarchive that's showing that even in cell culture, we can't translate the methods that have been used for making neurons from astrocytes have been largely demonstrated in brain astrocytes or brain-derived astrocytes. When you get into spinal astrocytes, they are very different beast. And so the same reprogramming factors doesn't result in the same level or types of reprogramming. So we're trying to first optimize that and hopefully along the way be able to start making specific types or subsets, if you like, of these interneurons.
Some topics have come up that also are questions on Zoom. So let's take a series of Zoom questions. Some of them deal with the endothelial issue that you were just talking about. One question was, is there any evidence that the endothelial cells actually form vessels that get perfused? Is there actually blood supply?
Right. So we've begun some tracing studies to see what sort of so injecting traces into the bloodstream, so we can use that to look at the distribution of these traces throughout our newly formed vascular architecture. But even just looking at the anatomy with the new vessels that we have formed through the injury site, there are red blood cells coursing through those vessels, which comprise human endothelial cells, which would suggest that there is perfusion.
Armin Blesch asked whether complimented you on your talk, and then wanted to know whether co-transplantation with the endothelial cells helps in the survival of the neural stem cells and neural progenitor cells.
That's an excellent question for which there's a rather complicated answer. And I think from the early studies, what we're seeing when we transplant these cells in combination, we're probably even further changing the types of cells that are being transplanted. So we might be shifting the population of neural progenitor cells based on what we transplant them with. And that could be, for instance, that there's cues coming from endothelial cells which drive the development or maturation of our neural progenitor cells into certain subsets of cells. That's something we're looking at because it certainly could be a complicating factor.
This question's more broad, and it gives you an opportunity, I suppose, to add anything to the endothelial cell contribution. This questioner wants to know, is there anything else you want to add as to how the endothelial cells are contributing to spinal cord repair other than what you've already
Not really.
about?
I mean, it's still early days for those studies, and like I say, it's an ongoing collaboration with a company, and so we have to work with them to make sure we can learn as much as possible about the cells and how they work in this system, and potentially be used for neural repair across other types of central nervous system damage or injury. It's early days.
Okay. And then this questioner, this gets back to the doxycycline treatment. What percentage do get converted to neurons?
So when in the in vitro studies we've begun looking at so far, we're looking at conversion of around 70 to 80%, which I think is, initially people thought that's not good enough. We want 100% conversion, which I disagree with. I think if you have a population of glial cells, we don't want to convert them all suddenly into excitatory neurons either. So maybe converting subsets into neurons is kind of ideal. We don't know what the perfect number will be yet for converting transplanted cells, but I would think incomplete conversion is probably a goal.
Thank you for your talk. I had a question about the first part of your talk. So I was curious if much is known about what triggers the recruitment of the V2A neurons or the V0Cs after the injury. Do we know if it is motor neuron homeostasis? Is it sensory information? And is there anything known about like what the biochemical pathways might be that might trigger recruitment of these excitatory neurons onto the phrenic motor neurons?
Right. So when we're looking at changes in connectivity, you mean after injury? So the spontaneous changes we see after injury, we don't know what drives that. But my guess is because these cells are connected normally, these existing connections, because there's been an injury that results in respiratory compromise, the spared networks end up working at a higher level. So these spared substrates, the activity is increased within those spared substrates. You do see a change in growth factor expression. For instance, BDNF can increase spontaneously. We think that a lot of these changes in the spared pathways rely heavily on these changes in growth factor expression, which may be changing therefore synaptogenesis, which is something that hasn't been as well studied within these subpopulations of cells. But certainly glutamatergic inputs onto the motor neurons that have been denervated, they increase. And so those glutamatergic inputs could come in part from even descending axons from the brainstem, but we think they're also coming from these excitatory neurons locally. That's something that remains unexplored so far, but I think the mechanisms are largely spontaneously driven by these varying changes in activity and perhaps related growth factor expression. And maybe that's why there's some variability between every animal. Relying on this spontaneous plasticity is not an ideal approach. And if we pair rehabilitation with that plasticity, we can certainly enhance that connectivity, and that makes more sense.
Yeah, sorry if I might ask a follow-up. So
Yeah.
Has it been similar? So something similar has been done with like the cervical and lumbar parts, right, where you can remove proprioceptive information and, the recovery is compromised. I was curious whether similarly in the phrenic motor system, is the loss of sensory information reduce the ability to recover? Or I'm just trying to figure out how the network knows that there has been an injury in terms of like activity, I guess. It's kind of that.
Well, the network knows because with an injury, if the respiratory system's compromised, there's a drop in oxygen saturation, and that's it's a chemical signal back to the brainstem that signifies an increase in respiratory activity in whatever's left. And so that certainly feeds directly back into the brainstem, which is spared by a spinal cord injury. And that increased descending drive over time, it certainly activates everything that's left immediately and tries to adjust for the fact that there's a degree of spinal shock. As that spinal shock subsides, that prolonged increase in activity is going to drive greater connectivity, and that increased connectivity will lead to this additional plasticity. Does that make sense?
And the sensory input, do you think, has there been an experiment where people have ablated the sensory inputs to see how that alters recovery?
We do. In terms of the spinal interneuronal populations we're looking at, we don't know what sensory input they're receiving. So it's a very interesting question, and that's a subset of studies that I wanted to look at was even just looking at primary afferent innervation of these interneurons. And as a collaboration with a group, David Fuller's group down in the University of Florida, we began looking at that some time ago and haven't had the chance to do follow-up studies. But that would be interesting to look within these specific subsets of interneurons, how that's changing over time. We don't yet know.
Great talk, Michael. Yet another question about the novel vasculature. I don't know, we're all sort of excited about this. It's a new set of thinking. And that regards both, my question regards both host-derived novel vasculature and graft-derived novel vasculature. Have you looked at the leakiness of these new blood vessels? Because, anecdotally, I'm hearing from people that when we do see these blood vessels formed, they are of a rather different quality than in the intact subject. And that would be, I guess, I don't know if you would call it leaky if you've just got blood cells leaking out, but of course you've also got to worry about the BBB and the formation of the BBB around those new blood vessels. Absolutely. Have you looked at any of that? Is that on the... That's
part of the tracing studies that we're doing. So actually some of my work eons ago when I was in Melbourne was looking at blood-brain barrier development. And so one thing to consider is as these new vessels form, there is going to be a permeability across those newly developing vessels that is not like the mature central nervous system. Following on from the fact that if there's any degree of persistent inflammation, that will also change the degree of permeability. That doesn't necessarily mean that they are, quote unquote, leaky. I think that difference in permeability is a response to the surrounding environment. Providing that there is not a massive extravasation of blood cells into the injury site, then it's not a problem. And we certainly do not see that. We don't see blood cells throughout the injury site. So I think if there's any change in permeability, it probably more closely tracks with that of developing vessels that are growing across that injury site. And as we do these tracing studies, as people have done in the past, these biotinylated different size molecules will show us whether these different sizes of molecules will pass into the lesion cavity.
This questioner wants to know, for practical and immunologic considerations, what are the prospects of having an off-the-shelf donor cell population?
So ideally, I think in terms of cell transplantation, we'd like to have an off-the-shelf population of cells that can be used. And as we move towards the use of human iPS cells to develop these new populations of interneurons and non-neuronal cells for transplantation, that's my research team's focus, is we think that these human iPS cells might be an ideal way to look towards that, and that may address some of the immunological concerns. However, many studies and ongoing clinical trials, even with human iPS cells, have shown that with early immune suppression, you get significant cell survival, and that immune suppression can be removed and you still retain some of these cells. That's been done in the use of iPS cells for treating Parkinson's disease, and I think that these results from that type of work will teach us a lot about how these same approaches can be used for treating spinal cord injury. So we're learning a lot from ongoing clinical studies in that regard.
Thanks so much, Michael. A great way to kick off the new academic year, medical academic year. Thanks, everybody.